Testing. Oh, there you go. So the neat thing about having a point of land named for you, well, there's two things. One is I'm not dead, and the other is I will never be pointless. So I will never forget the first time I put a dry suit on, and I slipped through 8 feet of sea ice through a hole about 3 feet in diameter into water that was minus 1.8 degrees centigrade. My heart was pumping, the adrenaline, but when I came out below the ice and I could look down and see the bottom as if I could touch it 100 feet below me, and I saw that the seafloor of Antarctica was covered with sponges and soft corals and starfish and sea urchins as far as you could see, I was in heaven. And for 10 years, I lived and worked going to a station called McMurdo, which is the biggest U.S. station that we have. It's below New Zealand. And there I studied all aspects of marine invertebrates, their reproductive biology, their development, and mainly their chemical ecology, how they produce chemistry to defend themselves from predators and these kinds of things. And every now and then in the cafeteria, somebody would say to me, you know, there's another station. There's a station called Palmer. It's only 44 people. It's perched on a point with a glacier behind it on the western Antarctic Peninsula. It's not like McMurdo with its 1,000 people during the summer. And I used to dream about getting to Palmer Station, and my science took me there. But what I didn't realize 20 years ago when I arrived at Palmer Station, that I was moving to an area of our planet that is changing very, very rapidly. And that's really what I want to share with you today, is what it was like to move to this part of Antarctica that's warming so quickly, and how that change is affecting it. So if you go back and you look at the old National Geographic that came out in 2006, you can see that as we began to study the Earth's warming, the focus was primarily on the Arctic, up north. And you can see the orange is really only sort of centered around where I live and work now at Palmer Station down on the peninsula. This was a little premature. Scientists hadn't quite really parsed the data. They hadn't gotten up into the satellites and looked at the information, and they hadn't looked at all the ground station data. And it turns out that there's really more warming in Antarctica than just the peninsula, that 800-mile length of land, but also the western portion of the continent. Now, you realize that Antarctica is a continent the size of India and China combined. This is a massive continent, okay? But it seemed to be mostly sort of a Western thing, the warming. But now we know that the Antarctic Circumpolar Current, the largest current on our planet that circles Antarctica in a clockwise direction, is warming. And that means that the ice sheets, these 1,000-foot thick layers of ice connected to the land, are beginning to melt not just on the western side of Antarctica, but also you can see the red dots over on the eastern side. So the east side of Antarctica is not out of the picture when it comes to warming. Well, how much warming are we talking about? Well, how fortunate that 20 miles from where I work at Palmer Station, there's a Ukrainian station, it used to be British, where they have been taking air temperatures every day for over 60 years. And if you look at those temperatures over that period of time, and look at the midwinter air temperatures, they've increased about 10 degrees Fahrenheit in 60 years. That is remarkable. That is the kind of warming that we hear about in the Arctic, maybe even more so. So what does that mean? It means that when I arrived at Palmer Station 20 years ago, about once a week, I would hear a huge crash, and I would leap up from my chair, and I'd run down the hall, and my colleagues and I would crowd around the door, and we'd look into the bay, and there'd be a big wave coming by because the Mar Glacier behind us, our neighbor, had calved. A big chunk of ice had fallen into the bay. It was very exciting. When I was at Palmer a year and a half ago, this was happening a couple times a day, not once a week. The sounds of the glacier calving have become normal background noise at Palmer Station. 87% of the glaciers along the western Antarctic Peninsula are in retreat. They are receding. Now, we have more quantitative data than running down the hall. We have a scientist who once every few years puts a backpack on, puts a GPS unit into the backpack that's talking to a satellite, and goes for a hike. And this individual, she hiked the edge of the glacier for me about 2 years ago, recording her position as she was hiking along the edge of the glacier, of course, to an accuracy of about an inch. And she came back and added another line to the map. And you can see where the edge of the Mar Glacier is now in 2017 and where it was back in the 1960s and 70s. And that little inset picture with Maggie and Chuck Amsler, my colleagues in it, represents when they first arrived in the mid to late 70s, when Maggie said you could open the door and just about step onto the glacier, and now you have to hike a half a kilometer to get to it. You can see it in this aerial image taken of the Mar Glacier. And look where it was in 1975 in the yellow line and where it is in 2013. So this is a very dramatic change in glaciers. We also have the capability now, if you can imagine the technology involved, in an instrument aboard a satellite that can measure glacial thinning or melting. And those red areas are areas where glaciers are thinning or melting. And this, again, is accurate to about an inch, so it's very accurate. And you can see that the melting of the glaciers is largely in the western portion of Antarctica, as I talked about earlier. And look at Greenland thrown in for good measure. Look at how the glaciers in Greenland are melting, are thinning. Well, this was a surprise. Nobody, scientists included, expected to find that the Thwaites Glacier on the western Antarctic Peninsula was receding at a rate that was unprecedented. An absolutely amazingly rapid recession. And this caused quite a stir in the scientific community because it was well known that the Thwaites Glacier is a figurative cork. It is the location where the ice sheet that sits on top of the western portion of the continent flows towards the ocean. That's right, we have 2 to 3 miles of ice sitting on top of the entire continent of Antarctica, and it's moving. On the west, it moves to the west, on the east to the east. And on the west, it flows through the area where the Thwaites Glacier is. The concern is that if the Thwaites Glacier disappears, breaks up, recedes, melts, that the rate of flow of that ice on top of the continent will be accelerated into the ocean. And this is concerning because when ice comes from land and enters the ocean, it contributes to sea level rise. And you know all about that here in Florida. You know on high tides in Miami, you can have seawater in the streets. So this is something that scientists are using to gauge how much sea level rise we're going to see over the next century. Will it be 1/3 of a meter or 3 feet? We don't know. But Antarctica is part of that equation. These are ice sheets. This is a map of the ice sheets that have broken out along the peninsula over the last 30 years. Ice sheets, to remind you, are about 1,000 feet deep and attached to land. And I want to show you a few of the breakouts. This is one called the Larsen B ice shelf over on the eastern side of the peninsula, which was imaged from a satellite on January 31st in 2002. You can see the striations in the ice sheet in this image, those lines. They came back a month later and the Larsen B ice shelf was disintegrating. 2 weeks later, it went out to sea. The entire Larsen B ice shelf. This is a piece of ice the size of the state of Rhode Island that floated away. And more recently, in a lot of the news, you heard perhaps about Larsen C and a big crack that had formed in the ice. And 2 summers ago, a piece of the Larsen C, the size of Delaware, floated away. This is when it did float away, it was the 5th largest iceberg that humanity had ever witnessed breaking out. And then finally, the Wilkins ice shelf at the base of the peninsula, a chunk of ice that has been breaking up for a period of, maybe at least 10 years now. It's still sort of dangling to an island, but when it breaks up and goes out, we're looking at a piece of ice the size of Connecticut. Now, the good news is that when these massive ice sheets break up and melt, they do not contribute to sea level rise. And that's because the ice is already in the ocean. Ice that's in water that melts doesn't contribute to rise. It's the same physics as a glass of ice water. When the ice melts in your glass, the water doesn't come over the top of the glass. So that's good. The concerning news about losing these ice sheets is that they're barriers. They're barriers to the flow of ice off land and into the ocean. And measurements indicate that the acceleration can be about 4 times greater when you lose the barrier of the ice sheet. And that does contribute to sea level rise. So those are some of the things that are happening with the ice, with warming on the peninsula. What about the marine life? What about the amazing community of marine organisms that live on this 800-mile-long peninsula? And that's what really has my heart. This is the community that I've been studying and working with the last 2 decades. Probably the most poignant story I can share with you is the story of the Adelie penguin, the penguin that you're looking at in this picture, the one that looks like Charlie Chaplin, the one that has a little kind of tuxedo on. And Bill Fraser, who is the gentleman in the back of this National Geographic picture, is a good friend of mine. Bill came to Palmer Station about 46 years ago. And for his doctoral dissertation, he tagged 16,000 breeding pairs of Adelie penguins. Now that is a doctoral dissertation. Bill, to his credit, has come back for 46 years, every year, to follow the fate of his penguins on this small cluster of islands right in front of the station. Now, if we look and see what's happened to Bill's penguins, it's not a good story. The purple line are Bill's Adelie penguins. And you can see on the left side of the graph, when that purple line started, there were the 16,000 breeding pairs. And Bill told me in an email about a month ago that this year, 1,100 pairs are left. So about 90% of the Adelie penguins have disappeared. And we know that they're not moving because they're tagged. They don't show up in other colonies, so they actually are disappearing. Interestingly, in the 70s, a small population of chinstrap penguins, the one in the right photograph on the top, showed up. And there's now, somewhere around 200 to 300 breeding pairs of chinstraps. And the Gen 2 has just arrived in force, starting in the 90s. There's now over 1,000 breeding pairs of the Gen 2 penguin in that middle picture. The Gen 2 is the largest of the 3 brush-tail penguins that you're looking at in this picture. Now, what's interesting is that the Gen 2 and the chinstrap really aren't part of the normal scene here. They're warmer weather penguins that, as the peninsula is warming, are moving down and extending their range into an area that before it was just too cold for them to operate. So they're moving in. Well, what's happening to Bill's Adelies? A couple of things. Probably the most significant is that, ironically, as the air is warming, it's getting more humid. With humidity comes snowfall. With more snowfall comes a problem. The Adelies are genetically hardwired to show up the same time every year and lay their eggs. And then along comes this unseasonably late snowstorm, covers the colony in snow, and when the snow melts, the eggs can't breathe in that water, and you lose a generation of penguins in a single snowstorm. And these are becoming increasingly common. So Bill thinks that this is a big problem. The other thing is the sea ice. And I haven't mentioned sea ice yet. Sea ice is only about 3 to 5 feet deep. And in the winter, Antarctica doubles in size because of the sea ice forming around the continent. And then it disappears and breaks up and goes away in the summer. The sea ice is so predictable that animals in Antarctica have evolved to be sea ice dependent. Their ecology is intricately related to the formation and the presence and the duration of the sea ice. The Adelie penguin is one of those species that's sea ice dependent. It uses the sea ice as a platform. The Adelie toboggans on its stomach across the sea ice, miles and miles, to get out to the edge of the ice and then enter the water where it's close to the krill, the populations of shrimp-like animals we call krill, that are the quintessential base of the food webs of Antarctica. And the penguins now have to swim much further to get to the krill populations because the sea ice is receding. So that's another thing that's putting pressure on the Adelie. Those other 2 penguins that I showed you are not sea ice dependent. Now, the krill themselves are also tied to the sea ice, and this is a fairly recent discovery. These krill, when they're teenagers, live underneath the sea ice, feeding on little plant cells called diatoms. And as the sea ice disappears, they're losing this resource of food, of refuge that the teenage krill are using. So this could put an added pressure on krill. You don't want krill to have a problem in Antarctica because krill are so important to so many organisms. For example, the baleen whales that feed on krill to a large extent. These humpback whales that happily are rebounding in large numbers in Antarctica from the days of whaling. On my most recent trip to Antarctica, I counted 76 humpback whales in a single afternoon. It's amazing. And if you're very fortunate, you come across humpback whales that are bubble feeding. There is nothing like standing on the bridge of a ship and looking off the side and seeing two humpback whales blowing a cylinder of bubbles underwater. And what they're doing is they're scaring the krill into a tight bunch within that cylinder of bubbles. And then the whales take turns diving down and coming up through the krill with their mouths open and coming out of the water, and you can watch the krill being sieved off the baleen right in front of you. And people are yelling, and it's just a phenomenal thing to see. So the whales are doing good for now. What about other species that are dependent on the sea ice? Well, the Weddell seal, one of my favorite seals in Antarctica, beautiful seal, has an amazing story of adaptation. The female Weddell seal has ice-chipping teeth. Only she does. The male does not have ice-chipping teeth. She has ice-chipping teeth because when she's pregnant, she swims up under the sea ice and looks for a weak spot, and then she chips at it until she can get a little breathing hole, and then she can take her time and continue to chip away with that hole until it's large enough that she comes right up through the hole and gives birth laying next to that hole. Her pup's completely safe from the ice edge where the leopard seals and the killer whales are prowling. And it's all because of that dentition. It's really an amazing adaptation. Now, I'm not so convinced that the Weddell seal is going to disappear with the sea ice issue. I think it may follow the sea ice, but we really don't know that. And what about the leopard seal? What about the top predator in Antarctic waters, the one that when we see and we're diving, we drop a siren into the water and we bring the divers up immediately? When the divers hear the siren, they know a leopard seal's been sighted from the boat above, and they come together back to back. By then, the leopard seal's probably making passes by the divers. Leopard seals are very territorial and very aggressive. And what we do is we wait until the leopard seals sort of moved off, and then the divers come up very quickly, and we pull them into the boat without doing any of the usual sorts of things to get them into the boat, and then we move to another area. Because we're scientists, and we don't want to test the hypothesis that the leopard seal might want to eat us. So, leopard seals, don't let me taint you, are beautiful animals. 1,000 pounds, 10 feet long. A leopard seal has never, ever, ever been seen giving birth anywhere except on the ice, on an ice floe, on the sea ice. So we don't know if leopard seals, as the ice disappears from the peninsula, are going to go to shore for the first time ever and give birth on land, or they will have to follow the ice. We just don't know. What about the small things in the water? What about the phytoplankton, the little plant cells, or the zooplankton that eat the phytoplankton? How are they being influenced by these changes in the environment? Well, we're very lucky. We have the NSF LTER, Long-Term Ecological Research Program, that's been operating on the peninsula for over 35 years. And the fellow in this white hat, Hugh Ducklow, is the director of that program. There's about 20 scientists that get together, and they get on a ship every year, and they take the ship on these transects along the peninsula, sampling everything: water chemistry, water physics, phytoplankton, zooplankton, you name it. It's about 6 weeks that they go to sea, and they've done this for over 35 years. Can you imagine the amount of data that represents, the amount of information? So I asked Hugh to do me a favor for my talk. Could he put all that in one slide? So I think he did a pretty good job. What you're looking at here is the northern tip of the peninsula in the 2 slides on the top, and the southern portion of the peninsula in the two cartoons on the bottom, okay? We're going to look at the top first, and we're comparing the 70s and 80s with the 90s and 2000s. And in the biggest of all pictures, what they're finding is that the peninsula is changing from a dry polar climate to a warmer, more sub-Antarctic, sort of humid climate. And the weather's changing. The weather is changing dramatically. It's cloudier and it's a lot windier, and the wind is churning up the surface of the ocean and pushing those phytoplankton down where they don't get enough sunlight, and some of the phytoplankton species that the krill like to eat are becoming scarce, and the krill are becoming scarce, and they're being replaced by salps. Nobody likes a salp. Salps are little tunicates. They're little gelatinous organisms about, in this case, the size of a walnut. And as the salps move in, you're losing a wonderful food source in the krill. Because if you think of a salp, think lettuce. And when you think of a krill, think steak, okay? So it's not a good exchange. Further down the peninsula, the changes aren't as dramatic, but they are changing, and eventually will probably look like the north. Now, what else is happening as it warms? Well, did you know that there's forests in Antarctica? All you have to do to be in a forest in Antarctica is to put your dry suit on with me. We'll slip into the water in front of Palmer Station, and we will be swimming amongst 150 species of seaweeds. Some of them are as tall as the roof above your head, with blades 10 feet long. It's a spectacular forest. I think the forests in a warming peninsula are actually going to do fairly well because the sea ice is going to be gone. They're going to have more light. They're going to grow deeper, probably, than they have before. They're probably going to extend their range further south. But we're also a little concerned because we know that these seaweeds produce chemistry that's toxic, that keeps things from eating it. And we know that these toxins are important in sort of structuring this community. In a future world with more light, you have more energy for the plants to play with. They might change the chemistry they produce or make more of it. It could influence the community dynamic. We don't know. What about the larvae? What about the offspring of starfish and sea urchins and tunicates and all these animals that are so diverse in Antarctica? The larvae in Antarctica are unique. They take a very long time to develop. They can take 3, 4, 5, 6 months to develop in the water before they settle to the seafloor and become a little starfish or become a sponge. The larvae that you're looking at is a sea star, a starfish. Now, we know from work that we've done and others have done that if you bring these larvae into the laboratory in Antarctica and you warm the temperature of the water 2 degrees centigrade, you warm it up 2 degrees, that they don't develop in 4 or 5 months, they develop in 1 or 2 months. In other words, they're extremely temperature sensitive. They haven't seen temperatures change like this for thousands, tens of thousands of years. Who knows? They just don't do the kind of response that you'd expect at that temperature. Now, developing quickly could be good in the sense that it means you're in the water less time to get eaten by a predator, but it could be a concern for feeding larvae. There are a lot of larvae in Antarctica that are feeding on phytoplankton. And phytoplankton in Antarctica bloom at a very specific time. What if they show up at the cafeteria and the door's locked? What if the phytoplankton bloom hasn't happened when they're ready to feed? And this offset of predator and prey is what climate scientists are worried about in all of our ecosystems on the planet, not just in Antarctica. Now, there are some other species besides the Gen 2 penguin and besides the chinstrap that are moving in. They are extending their range as we rapidly warm. There didn't used to be elephant seals like you see in the left-hand top picture. Up to 2-ton elephant seals living near Palmer Station. Now you can hear them barking when you walk out the door, and you know they're having pups. They have moved in. They have established a breeding colony. And I've been told, and I haven't been there to experience this yet, but if you're there in the early part of the summer, on a warm day, there'll be 10 of them laying in the middle of the station. Do you know what that smells like on a warm summer day? And fur seals, these beautiful seals that were almost hunted to extinction for their fur, are moving in. from warmer areas. They've extended their range. They haven't started breeding near the station, but I suspect they will. And probably the most poignant and dramatic example I can give you of a range extension, or maybe an invasion, I'll set the stage with this slide. You are looking at the seafloor community of Antarctica on the shelf. This is a very ancient seafloor community. It was established 40 million years ago when Antarctica separated from South America and allowed the establishment of the Antarctic circumpolar current that effectively locked Antarctica into the deep freeze. Species that could adapt to the cold did, and many did not. There are some very conspicuous things that are not in Antarctic waters. There are no sharks in Antarctica. There are no fish with strong, crushing jaws. All 250 species of Antarctic fish are wimps. They have weak little mouths. They chew on little crustaceans. There is nothing in the Southern Ocean with a claw. There's nothing in the Southern Ocean that surrounds the waters of Antarctica like a crab or a lobster. So you can imagine the shock of my colleague Sven Thatchy, who on January 25th, 2007, was sitting in a ship, flying a remotely operated submarine below him, 1,123 meters below him, when for the first time ever, scientists came across king crabs moving up the Antarctic slope. Now, we knew that king crabs lived in the deep sea around Antarctica for a very long time, but they were never seen on the slope or the shelf leading up to the continent, because it was thought that at very low temperature, king crabs have a hard time. If you put king crabs at a low temperature, they act like they're drunk. They fall over. They can't feed themselves. They miss their mouth. Effectively, what happens is there's magnesium in their blood that acts as a narcotic at low temperature. And so we thought they were being kept at bay. So what could be happening, and we have data to support it, that this deep water surrounding Antarctica is beginning to warm. And that curtain, that physiological curtain, may be opened, and king crabs may be moving in. Well, I wrote a grant proposal to NSF with a colleague to see if we could document how many king crabs are on the slope. Have they gotten to the shelf where a crushing predator with claws could do a lot of damage to that community? Antarctic species are very poorly defended. You could pick up an Antarctic clam and crush it with 2 fingers. The shell's very thin, and probably because they've never dealt with crushing predators. So I wrote the proposal with a colleague in Florida, Rich Aronson, at FIT. We sent it off, and in the meantime, while we're waiting, another group of scientists bumbled into, 1.6 million king crabs on the Antarctic slope in the Antarctic, a canyon called the Palmer Canyon Deep, within sight of Palmer Station. So they're poised. So we got our grant, and we've now taken a ship to Antarctica 3 times. And what we do is we rent a submarine from Woods Hole, and we tow the submarine behind our ship in 10-kilometer-long transects up the Antarctic slope and onto the shelf, taking pictures of the seafloor every 15 seconds. And we do this at various points along the shelf and slope. You can imagine the amount of data we have to analyze, to look at all those images to see if the king crabs are there, and are they breeding, what are they eating, are they causing ecological issues. When you have that much data, you turn to something very critical to scientists called the graduate student. And we have had many graduate students analyze tens of thousands of images, and basically the king crabs are there in large numbers by the millions. They have not reached the shelf yet. They're still several hundred meters below the shelf, but we hope to follow that story. And last but not least, let me mention the stepchild to climate warming. We call this ocean acidification. It doesn't get near the press that it deserves. 30% of the carbon dioxide that we've allowed into the atmosphere since the beginning of the Industrial Revolution has effectively found its way into the world's oceans. And as those oceans absorb carbon dioxide, there's a chemical reaction that takes place that lowers the pH. In other words, the acidity of the water has increased. It's increased about 30%. since the Industrial Revolution. And that means if you make a shell or you have to maintain a shell, you have a challenge. And Antarctica is the canary in the coal mine for ocean acidification for several reasons. One is, I've already told you, things are very weakly calcified in Antarctica. There's not much of a shell to begin with. And secondly, the colder the water, the more CO2 is absorbed. It's just a simple physics relationship. So we have the Southern Ocean, which is probably the biggest depository or repository for CO2. So we actually have had some grants to look at ocean acidification in Antarctica. This is some work that others have done, not us, but is very striking in that it makes the point that you can go to the Southern Ocean today and take a net and dip up one of these beautiful little sea butterflies. This little sea butterfly has evolved to leave the seafloor and it has little wings. It flies in the water, and they're as common as the stars in the sky. They're as small as the tip of a pencil, but because of their abundance, they're part of the global carbon cycle. You can find an area in the Southern Ocean today where these little animals, under a microscope, are showing evidence of dissolving of the shell. The aragonite shell is etched by ocean acidification. This isn't your grandchildren seeing this. This is happening today. So we have had several projects where we've been looking at calcified shelled invertebrates and ocean acidification. This is Palmer Station, where the work's being done. This is where I'm headed in a few weeks for my next field season. And it is a wonderful place to do marine biology. The one thing the U.S. government doesn't want to do is to get you, as a marine biologist, all the way to Antarctica and not make sure that you can do your science. So we have a wonderful science staff support group at the station. Everything that we need is taken care of. We've meticulously ordered everything before we get down there. It's a wonderful place to work. Now, I want to tell you a quick little story because one of the species we found to be susceptible to ocean acidification is called an amphipod. It's a little crustacean, maybe a few, half an inch long. This one we haven't tested yet. But I was out on the ice at McMurdo Station in my early career, and I looked down into a dive hole and saw this little amphipod carrying an orange backpack. I grabbed a net, got it up, took it to the marine lab, and there was a fish biologist, John Jansen, there, and the two of us launched a project to find out what in the world this orange backpack was. Well, we pulled it off the back of the amphipod, and it opened its wings, and it fluttered away. It was a sea butterfly. It belonged to a group of sea butterflies that don't have a shell, a naked sea butterfly. And so we put nets out into the Sound, and we found hundreds of these little amphipods carrying sea butterflies. Why would they go to all the trouble? So we brought them in the lab, and being experimental scientists, we did the experiment. We took the sea butterfly off the back of this little shrimp-like amphipod and we offered it to a fish, and the fish would eat it. They loved this little amphipod. And then if you left the little sea butterfly on the back of the amphipod and you offered it to a fish, they took it in the mouth and then spit it right back. And away went the little amphipod carrying its sea butterfly. It was one species abducting another species for its own defense, because in that sea butterfly was chemistry that tasted terrible to fish. So it was a very smart amphipod. And it was such an unusual relationship that we were able to publish this in a journal called Nature. So that was very exciting. So this is a list of what I've talked about. It's quite remarkable. What's happening to the ice? What's happening to the glaciers? What's happening to the animals that are dependent upon the sea ice, like the Adelie penguin? What's going on with the forests? What's happening with species showing up that haven't been there in the past as they extend their ranges, maybe even king crabs, ocean acidification? This is a remarkable change. This is a community that for millennia has looked like this, where you have very predictable sea ice formation every winter. You have a very stable population of Adelie penguins. You have krill. You have a seafloor community. You have the forests, and you have the Antarctic circumpolar current, the ACC, which is high in nutrients feeding this ecosystem. And look where we're headed. Probably by mid-century, the sea ice won't be there, the Adelie won't be there, the Gentoo and the chinstrap will have moved in, the krill populations will be much lower, we could have king crabs, the forests will have expanded, there'll be more salps, and again, all being fed by the Antarctic circumpolar current. Now, you might be sitting there right now thinking, what's the big deal? Communities change all the time. They're very dynamic. But the big deal here is the rate of change. When people ask you about what's the big deal about climate change, it's not that climates don't change. Of course they do. It's the rate that they're changing right now that has biologists so concerned. Organisms are not able to make these changes necessarily. Not all of them. And some of the key players are disappearing. So this is very important to think about. So why should we give a darn? Antarctica, that's a long way. That's a long way from Whitney Marine Lab. It would take us a week to get there if we went to the airport right now. Minimum. And that's if the ship is waiting for us when we get to Punta Arenas. Out of sight, out of mind. So I want to give you an example of one of the things that makes this story a little more realistic to you, perhaps, and valuable. And that is, as part of our chemical ecology work in Antarctica over the last 3 decades, we also have a drug discovery program that's aligned with our chemical ecology work. And so when we're diving at McMurdo Station under the ice, we're collecting material to extract and see if these chemicals can fight cancer and AIDS and cystic fibrosis and different kinds of human diseases. Same thing at Palmer when we're diving out of our Zodiacs. And we found some pretty neat stuff. We found a tunicate, this orange blob, produces a compound called palmarolide. We named it palmarolide after the station. We sent it to the National Cancer Institute. They screened palmarolide against 20 different types of human cancers, and they called us up. And they said, we're really excited about palmarolide because it's very potent and it's very specific. It's only active against one type of cancer, and that's what we're looking for. Can we work on it? And when the NCI, the National Cancer Institute, asks you if they can work on your compound, you say, yes. Work on it. And they took it and they were able to make palmarolide in the laboratory because you are not going to take ships to Antarctica and harvest thousands of kilograms of this thing and come back across the Drake Passage. So they could make it in the lab. It took a fair amount of effort, but they did it. They were able to put palmarolide into a mouse, radio label the compound, and prove that the chemical could move across tissues and membranes and find its way into a little tube that had the cancer cells in it. In other words, it was deliverable to cancer cells in a living system. So that was good. And more recently, and perhaps most exciting, is that my colleague Bill Baker, the chemist I work with, has sequenced the gene that produces palmarolide. And now you don't have to make it in a laboratory. You could put it in an E. coli, a bacteria, and just have it make the chemical for you. So who knows? Someday we could have, from Antarctica, a drug that fights melanoma skin cancer. How cool would that be? And there's a need for that. We don't have many drugs that fight melanoma. We found a protein in an Antarctic red alga that's active against the H1N1 flu virus. And last but not least, we've also found, very excitedly, a compound that's active against MRSA. This is from an Antarctic sponge, and it's active against the form of MRSA, which is the bacteria you get now when you go to the hospital and they can't get under control because the antibiotics don't work. There's a form of MRSA that's under a biofilm. A biofilm is a layer of mucus and proteins and things, and if you have a surface like an artificial limb or a knee joint that's going in, it'll get biofilms on it. The MRSA gets under there, they can't treat it. They have to take out the inserted knee and clear you and do a second surgery. So they're excited because this is the first chemical compound that's ever been able to show activity under the biofilm against MRSA, and that's very exciting. So why am I telling you all this? In the context of climate change, what I'm saying is that Antarctica is a very ancient community. There's been tremendous amount of evolution of chemistry among these organisms. This is the tip of the iceberg, for finding new medicinal compounds. Why would we squander that with allowing the waters to warm and these species perhaps to disappear. Now, I lecture on climate all across our country, and I used to get asked this question: is the climate really warming? I have not been asked that question probably in 5 years. And you can go online and you can Google climate warming, global warming, whatever you want to call it, find lots of evidence. But I want to show you the data that convinced me that it's true. It really is. There is no way that anybody can take these data to task. In science, we call these hard data, right? So, the question that I get now is, okay, the planet's warming. Do we have anything to do with it, or isn't this just a natural cycle that the planet goes through every now and then, and we really don't need to worry about it in the big picture? Well, I want to show you the data that convinced me that we have a role in what's happening with this warming, okay? And how perfect that this data comes from Antarctica for my Antarctic talk, right? All right, don't get scared by this slide. What you're looking at is an ice core. An ice core is about the diameter of a grapefruit, and it goes down through the ice. And this one went down through the ice that sits on top of the continent of Antarctica. Remember I told you how thick that ice is? This one went down 3,600 meters into the ice sheet. And you're looking at the ice core like it's laying on its side, and as you go to the bottom of the ice core on the left, you're going back in time. At the bottom of the ice core, you're going back 420,000 years ago when that ice was formed. And you know what's really cool? There are little air bubbles that are trapped in the ice at that time that are representative of the atmosphere at that time on our planet. And you can analyze the amount of CO2, the greenhouse gas CO2, in our atmosphere at that time. And that's what that red line is you're looking at that goes up and down and up and down and up and down. And the blue line is temperature, which is calculated indirectly. I put a big black line on top of the red line to show you that in the past 420,000 years, there has never been more than 300 parts per million CO2 in our atmosphere. Look what happens over on the extreme right portion of the graph at the beginning of the Industrial Revolution. The amount of CO2 skyrockets. In fact, this graph is out of date. I think we're up to like 411 parts per million now. I mean, we have seen 25% increase in a well-documented greenhouse gas since we started burning fossil fuel. Now, you have to make up your own mind if that's a signature. I believe it is. I think 99% of scientists believe that this is real, and this is something we really have to consider. Now, you're wondering why that red line goes up and down in that 100,000-year period. That has to do with the elliptical orbit of our Earth around the sun in a 100,000-year time period. It's called the Milankovitch cycle. So, gloom and doom. But I'm not going to leave you with that today, okay? I'm going to leave you with a very positive message. And how great that the story I'm going to tell you right now happened in Antarctica. In 1985, 2 technicians were sitting at their desk at a lab not far from where I work. And they looked at the data and they said, our boss in Great Britain is not going to believe this data. And you know what happened? They were right. And he said to them, you're going back to Antarctica next year with a new spectrophotometer. You're going to do all the measurements again and tell me what you find. And they did. And they published in the journal Nature probably one of the most important papers of the 20th century announcing a massive hole in the ozone, that layer of our atmosphere that protects us from ultraviolet radiation that was so big, it was all over the continent of Antarctica. It was so big that it periodically drifted over New Zealand. And when I was on sabbatical on the South Island of New Zealand with my family many years ago, I used to read the newspaper with my coffee, and in the weather section, it would tell you what level of sunscreen to put on as a function of whether or not the hole in the ozone was over New Zealand. And it may be coincidence, but New Zealand has a very high level of skin cancer, the highest of any continent. So, what a discovery. Something that affects our health, something that can even affect climate, as the whole can, had occurred on a global scale. But what was so amazing to me was in 1987, 2 years after the Nature paper came out, 20 countries sat down around a table in Montreal, Canada, and ratified the Montreal Protocol that regulated the chlorofluorocarbons, the refrigerants that were destroying the ozone. And today there's 197 signatories to the Montreal Protocol. 197. It's probably the most successful global treaty of all time because it's worked. The hole in the ozone is actually stopped growing. Susan Solomon, the scientist who discovered the chemistry behind the loss of this ozone, told me that they now think it might close mid-century instead of end of century. How remarkable. And you know, the companies that made the refrigerants didn't go out of business. They didn't lose jobs. They used ingenuity, and they designed new compounds that don't destroy the ozone. So it can be done. So we can address these big global issues. Of course, CO2 is a bigger problem, but it does give us a model to think about. So before I finish, let me just tell you a little bit about my own story. Why a scientist who could happily be doing his research in his lab or in Antarctica and not lecturing across the country is doing this. I just felt it was very important as a personal witness to what's happening in Antarctica to share that story. And the first thing that people told me is, if you want to share the story, by God, you better write a book. And then I discovered to write a book, you have to have something called a literary agent. And then I found out that to get a literary agent, you have to have a book. So I figured out that if all else fails, turn to your connections. And so I was very fortunate that I had hosted E.O. Wilson, perhaps the most famous living scientist today, who had grown up in Birmingham and in Mobile, Alabama, and gone up to Harvard for 70 years. E.O. Wilson had sort of adopted me because when I nominated him to come to UAB and get our big award, nobody told me that I had to be his gopher. I had to pick him up at his hotel in the morning and get him to lunch with the dean of medicine and then get him back to his hotel. And by the end of the week, we were friends and colleagues, and he's mentored me ever since. And he knew I wanted to write, and he got me a literary agent. And when my book, Lost Antarctica, came out, I had no idea what doors would open. I didn't know that the director of the E.O. Wilson Foundation, Biodiversity Foundation, would call me and ask me to serve on their advisory panel. I didn't know that she'd say to me, Jim, we love your chapter on the Adelie penguin. We want to turn it into a 3-minute video that's going in all the zoos and aquariums across the country. It's a poignant story on climate. And we love the prose, and I thought she was asking me to narrate it. And when I said so, she said, no. She said, we'll have Harrison do it. And I said, Harrison who? And she said, Harrison Ford, of course. He's on the advisory committee with you. And I said, I had no idea. So Harrison reads from my book in a beautiful 3 or 4 minute. You can get this on YouTube, and you can listen to him reading about the Adelie penguin. He took this very seriously and spent a lot of time doing it. So I encourage you to check it out. My book also had sort of a connection with this day. Who would have thought that my science director, or the station leader, I should say, would come to me and ask me if I would host a visitor at Palmer Station? And when I asked them, of course I would, who is it? They said, I can't tell you. And I said, why? And they said, security. And I said, but we're in Antarctica. And so the next day, Bill Gates and his father, Bill Sr., showed up. And Bill's stepmother was there and his son, Rory. So I had the 4 of them for the whole day, and I took them around, showed them all the labs, and got to talk to Bill about his work in Africa and medicine. And Bill was very interested in climate change and renewable energies, and we kind of hit it off. And after lunch, it was my job to take Bill and his father up to the room with the big screen to show them the National Science Foundation video on the U.S. Antarctic program. So I had gone up there and put 2 big chairs in the front of the room for Bill and his father, and they were sitting in those chairs, and the IT guy came in and could not get Microsoft to boot up. Can you imagine what it must have been like to be that IT guy? And Bill's father looked at his son and said, You really need to fix this, Bill. And I thought, this is my Microsoft moment, yes. So anyway, Bill read my galleys and wrote a nice little book jacket for me on this, so that was very nice of him. He asked me if we had enough money, and I said, yes, I blew it. So another thing I do now is I take people to Antarctica, and I've done this for 13 years. I'll do my 13th cruise in a few weeks. It's called the Communicating Climate Change Cruise, and it's run by Abercrombie & Kent, a wonderful travel company. And they came to me and asked me if I would do this. And I was reluctant at first. I thought, tourism in Antarctica, I don't know. But I did it, and I fell in love with it, because the people that go on this cruise become ambassadors to Antarctica. They fall in love with the continent. They talk with their senators, their congressmen when they get home about climate change because they've seen its effects. It's a wonderful experience. We visit the station where I work, it's just marvelous. So if any of you are interested, find me afterwards and I'll give you a card and we can do something. The other thing I've done is I have been involved in taking climate change themes out in faith communities. And this is, I'm in the South. Religion is an important component of life. And I think this has a tremendous potential. And this past year, I was the keynote speaker for the Alabama Episcopalian Conference and also the South Carolina one, where I was speaking to 500 people. And many of them went back to their parishes and started sustainability groups. So I think that there's a lot of potential here as well, because the care of creation is certainly part of what needs to be done when you think about climate. I've also become the national face of the Can We Talk Climate campaign by the National Nature Conservancy Organization. So this is a pretty cool idea, a pledge with no money involved. You go to the webpage, you sign in, you make your pledge. Your pledge is that in 7 days' time, you will talk to one other person about climate change. And then on the website is all sorts of great information about how to talk to people about climate change, how to find common ground, how not to be dogmatic, how not to be political, how to talk about the issue, and you'd be surprised how effective that can be. I have to tell you a quick story. I was a big speaker for the Tennessee Aquarium, and when I went up to Chattanooga, they took me to the radio station the morning of my talk. And of course, you know the radio station, right? NPR, right? The scientist, he goes to NPR, right, to talk about climate. So I go to the NPR affiliate, and then my host says, you know what? I have a friend who's got a radio station out in the countryside, and he wants you to come out here and talk about climate change and global warming. And I said, okay. And we drove into rural Tennessee and this little radio station. And the guy who was running the station, he says to me, now this is a talk radio station, and when you finish talking about climate change, the phone's going to ring and it's not going to be pretty. And he was right. The phone rang and this guy was livid. I mean, I was part of a massive conspiracy of scientists that were trying to get rich and on and on. And finally, the radio host just grabbed the phone and said, Joe, stop. And then he said to Joe, you've been telling me that your tomatoes in your garden have been coming in earlier every year for the last 5 years. And those birds that you're seeing on your feeder you haven't seen before. And you know what? In a few minutes, they were talking climate change in Joe's backyard. And he was kind of coming around. It was so interesting to see, if you find that common ground, how effective it can be. And let me just finish now, I'm done, with one final story. This year I was honored by the Explorers Club to receive an award for my Antarctic research and outreach. They give this every 4 years. And to me, the honor wasn't so much being in front of 2,000 people in a big fancy hall in Manhattan. It was who was sharing the stage with me, the Apollo astronauts. I mean, how cool was it to have 8 Apollo astronauts listening to my talk? And how cool was it at the end of the night when they all sat there staring out at the audience, and each one was asked to tell a little story that had never been told before, and people would just love to hear it. And my favorite one was Mike Collins, who we had dinner with, right there. He's the one that stayed in the craft that circled the moon while the other two went down. Mike Collins did not like Buzz Aldrin. Buzz is a character. I met him. He really is. He's a handful. But he walked on the moon, and he knows it. Mike had a list for Buzz, a list of things that he wanted Buzz to change about his behavior. And when Buzz and Neil Armstrong came back to the mothership from the surface of the moon and asked, they have to professionally ask, can we reconnect? Mike said, no. Let me get my list. And he proceeded to go down the list, and Buzz had to, answer in the affirmative to each one of these requests before they allowed them to dock. And I just love that story. But the most poignant moment, and the reason I put Rusty's name over his head there at the end, Rusty Schweikert, who was the pilot of Apollo 9, stood up. Everybody else had been sort of casual sitting. He stood up, and the whole room became quiet. And he said to us that, all of us on this stage have been up in space. And all of us have looked back at our planet, and all of us know how fragile this planet is. If you look at how thin the biosphere around the planet is, and that's something we really need to think about. And I'll just add as a scientist that our Earth, we have a sample size of 1, okay? We have a sample size of 1, and we can't afford to experiment with it. And I'll leave it at that. Thank you.